Hello. Today is Wednesday, October 21st, and this is a recap of what we discussed in biology today. What we looked at was the plants in BTB experiment that was conducted in carbon time by Daryl and Nina. We first had to set up our experiment to understand what we thought might happen. So we have two drawings here, one for the light and then one for the dark. In both of these drawings, we have a closed container that contains a plant and two petri dishes containing BTB. Each dish has a different concentration of carbon dioxide. The blue BTB would be low in carbon dioxide or no carbon dioxide. The yellow BTB would contain carbon dioxide. In both situations are the same. Both setups are the same. The only thing that is being changed is this experiment down here is going to be conducted in the dark, and this experiment up here is conducted in the light. We asked people then to predict what did they think would happen when we let these experiments run over the course of 12 hours. A plant in light and a plant in the dark. When we were discussing these things, we were discussing what would cause possible changes. Would the plant absorb CO2 at the very top up here? Would it absorb CO2 from the petri dish, cause the petri dish to turn yellow? Or would the plant be releasing CO2 and cause this dish down here that is low to turn yellow? You have two different possibilities, either both turning yellow or both turning blue. And so we looked at that for both of those. So then in the video that we watched in class, which you can obtain also on our classroom site, we were able to see what happens in the experiment. So 12 hours later, we noticed that in the experiment with light, both petri dishes turned blue, which means that CO2 must have been removed from this petri dish and must be absorbed by the plant. So in this case here, the plant is absorbing CO2. Down here in this situation in the dark, both petri dishes turned yellow, which means that this petri dish absorbs CO2. Therefore, the plant must be giving off CO2 in this bottom picture here. Let's make those adjustments to those pictures there. And then down here, we're giving off CO2. So we made those observations, and then what your task is to explain what is happening and why. So you are assigned to draw a triangle that explains this. And let me show you what that looks like. So in your task, what you need to do is you need to draw two triangles, one for light and one for dark. I have the examples shown here. What happens to plants in the light? We add our phenomenon. We know that the BTB changed from yellow to blue. We know that CO2 is being released. Because of this, we want to know, well, what's happening inside the leaf? So we're going to zoom into that, and we have to explain from an atomic molecular viewpoint, how is matter changing in the leaf? And how is energy changing? We also need to come up with a formula and what the name of that formula or what it's called. That's the other task there. Notice that the formula is even given to you in this video. You need to then create another triangle in your bottom half of your paper so you can take a picture of this and put it in the Google Doc of what happens to plants in the dark. So we would have another triangle down here of what happens to plants in dark. Once you've completed both triangles, make sure you take a picture of those and submit those to insert them into your Google Doc, and then submit your document for today. Thanks, and have a great day.